The chapter that we're going to handle now is microbes in human welfare. Microbes. When I say the name called microbes, one important thing comes into our mind, which is common in everywhere in the laboratory. It is a glass material by which, or made up of lens, by which we can observe the tiny particles. What is that instrument called as?
Microscope, sir.
Microscope. Scope means vision, micro means small. So this microbes, what is this microbes? They are actually living organism only. The microbes is actually a living organism. So what is actually it is microbes in human welfare. So what is microorganism is the first question. These are tiny organisms which we cannot be seen with the naked eye. That we are learned in grade 8 and grade 9. What are microbes? We'll say microbes are tiny organisms which cannot be seen with the naked eye. Okay, fine. We all know that we are always living with living organism. We are always in interactions, and we cannot live without any interaction. Definitely, we are an interacting organism. The world can survive without human being, but human being cannot survive without the world or without the environment. So microbes, how we are benefited from the microbes. The microbes is not giving a benefit only for us. The microbes have some enzymes or microbes has some products. These are they're extracting some certain products from the microbes and using for our own welfare. So that's why we say what is biotechnology? Use of living organism for human welfare. That is called as biotechnology. So here, from living organism, it is also a type of biotechnologically oriented works, but a very simple work which actually we commonly know. The microscopic organism, which are not at all visible with the naked eye and viewed under the microscope, are called as microorganism, or commonly we call as simple term microbes, which includes bacteria, protozoa, yeast, microscopic plant, and viruses. The viruses, viroids, and prions, of course, we have learned, are proteinous infectious agent which also included under the microbes. So the common things that we have to discuss here is microbes. They are omnipresent. They are present everywhere, and they have a high degree of adaptability. They can live everywhere. So, where and all, since microbes are everywhere, how we are deriving the essential products from them? So, microbes in household products, in home, where and all microbes used, and what are the benefits that we are deriving from them? Microbes in industrial products, how microbes are used in industries, especially the fermentation industries. Of course, you all know sewage. Means the dirty water. How the microbes are employed in purification of the dirty waters, because we should not straight away discharge the dirty water into the sink, that is the river. We have to treat it and we have to send how it is treated, because the we throw everything into the environment which may control the hazardous material. It can be biodegradable or non-biodegradable, which are most of the things are medical waste, kitchen waste, and which are actually the paper pulp waste, office waste. So all this actually even human body waste, all these are nothing but the sewage water only, how it is treated. And this commonly, that gobar gas, how the microbes are involved in biogas production, and microbes as biocontrol agents. About biocontrol separately, I'll discuss. And you know that last year your seniors also have done a lot of research work on biocontrol agents, and we have our own biocontrol products, which is there in our laboratory, and microbes as biofertilizers. So this is the general instruction I have given you. This is called a streak plate culture, where we are culturing microorganisms. This is slant culture. This is nothing but the medium in a test tube in slant media where we'll inoculate the microbes. This is called plate culture, where we will be having a small petri plate where will drop the nutrient-rich agar media, and which help us to grow the microbes. When school starts, we can grow microbes in our laboratory also, and even I'll tell you how to identify the colony categorization. So microbes, that they are everywhere, and for a lot of benefits we are obtaining from microbes. So where and all it can be used, that is what the first thing we are going to focus. The first one is in household products. What is the shape of this bacterium, children? It's a bacterium. It is spirala or rod shaped?
Rod shaped, sir.
Rod shaped. So what is rod shaped called as?
Bacillus.
Bacillus.
Bacillus. So curd, production of yogurt or yogurt like products like curd are common example of fermentation activity of microorganisms. The common example is curd from milk. Microorganisms such as Lactobacillus delbrueckii, we say the genus is Lactobacillus, and which is commonly called as lactic acid bacteria (LAB), which grows in milk and convert it into a curd. So during growth, the lactic acid bacteria produces acids that will coagulate and partially digest the milk proteins, which is called as curd. So what is the role of lactobacillus mean? There are three important roles. First thing is they will convert the milk into curd. Second is in the curd, what they will increase mean it will increase the concentration of vitamin B12, psychobovalamine. So the lactic acid bacteria increases the concentration of vitamin B12. In our stomach too, the lactic acid bacteria play very beneficial role by checking the disease causing microbes in our gut. So drinking curd is very good for your controlling the microbial flora in your gut. Therefore, there are three important role of lactobacillus. One mark question for your board exam. Conversion of milk into curd. Second one, increases the quantity of vitamin B12. Third one, checks the disease causing microbes in our stomach. So the microorganism involving in converting milk into curd is lactobacillus. So we have lot of lactobacillus called Lactobacillus bulgaricus, Lactobacillus thermophilus, Lactobacillus delbrueckii. So all these names are not there in your book very clearly. So the common other microbes we're going to call lactic acid bacteria. So if you have some stomach issues with respect to the microbial flora concentration, we call lactic acid bacillus a tablet in the name of trade name of Sporlac it is given to check the disease causing microbes in your gut. Any doubt in microbes in household products?
No.
No. Second one is, microbes in household product. The common thing is a yeast. The first one is a bacterium, Lactobacillus. Second one is a yeast. It is called baker's yeast. The same thing is also called as brewer's yeast when we use for alcoholic fermentation. The same microbe called baker's yeast called as Saccharomyces cerevisiae. It is commonly used to make bread by fermenting dough. So numerous food items like idli, dosa, jalebi, and even bread, all are prepared by fermenting process, where large amount of CO2 is evolved. Because of the CO2, you can see small small holes in the bread and rising of the dough. I think if your mom will prepare the rice dough and keep it the night and early morning, you can see it will just will be very soft and it will actually come like a puffed appearance and little bit will be rise level will be raised because of large amount of carbon dioxide evolved during this fermentation. And next is toddy. You all know toddy is very famous in Kerala and fermented toddy is actually alcoholic. The toddy straight away, this is actually an alcoholic toddy. The toddy in Tamil we say kal, that is called as toddy. So it is actually a whitish fluid. Straight away from the source, that is from the sap of the palm, if you just take and consume, it is not alcoholic. It is actually very good for your health. So when you take here toddy, so toddy is made by fermenting sap from the palms. And even there are some microbes used to ferment fish, soya bean, bamboo shoots, and to produce cheeses also. And microbes are also used. It is the oldest method. You can see in Tom and Jerry movie, you could have seen that cheese means always there will be somewhat and they put lot of holes where Jerry will make a lot of will stay inside and it will enjoy. So with cheese they will fight. The cheese, so cheese will have certain flavor that is because of microbes. You can see the large amount of holes are there in the cheese. So large holes in Swiss cheese is due to the production of a large amount of carbon dioxide by a bacterium genus is Propionibacterium, species is Shermani. The Propionibacterium shermani, this microbe, they will produce large amount of holes, their size, and it will be very shiny also. The cheese will become so shiny and it will be very soft because of a thermophilic bacteria that is called as Propionibacterium shermani. In fermentation process, they will take the carbohydrate substance from there and they release large amount of carbon dioxide, and even they also ripen the cheese also, which gives a unique flavor, and large amount of CO2 is because of evolution. Means why they, in the board exam question they will ask you why Swiss cheese have large amount of holes? Because of large amount of carbon dioxide production by the bacterium Propionibacterium shermani. This is called Roquefort cheese, and it is also how we are going to means by a bacterium called as sorry, a fungus called as Penicillium roqueforti. By bacterium Penicillium roqueforti, it is not there in your book. The Roquefort cheese shows a unique greenish blue mottling, that is because of a specific fungi called Penicillium roqueforti. It is probably the best known cheese made from sheep's milk. It's Roquefort cheese. So these are the three important products, four important products. The first product is called curd, second is called dough making, and next one is your cheese making, and the fourth one is toddy. These are the four important products that we have discussed in household products. Now we go for the industries, and today we'll stop with the industries. Okay, so industries. If you know that a large number of powerful industries based on beneficial activities of microbes, which have been set up in recent years to produce a varieties of products in enormous amounts, like alcoholic beverages like beer, wine, and this whiskey, non-alcoholic beverages like curing of coffee beans and tea leaves, acids like lactic acid, citric acid, acetic acid, glutamic acid, and enzymes like streptokinase enzyme, vitamins, hormones, amino acids, antibiotic, vaccines, steroids, etc., etc. We can add lot of things we are actually taking from microbes. Even the human insulin called humulin is actually obtained from microbes, but genetically now they are able to produce by biotechnological method. Previously they are produced from slaughtered animals, then they shipped on to microbes, and microbes, even from that lot of vectors are created. About that separately we'll discuss. So production, the first production is in industries. The first product we're going to focus alcoholic fermentation or fermented beverages. There I used the yeast called Saccharomyces cerevisiae, which is used for bread making and commonly known as brewer's yeast. There it is known as baker's yeast, is used for fermenting malted cereals and fruit juices to produce ethanol. That means instead of going for lactic acid fermentation, these yeast will act on a carbohydrate substances, and they go for alcoholic fermentation. So wine and beer are produced without distillation. There is no such of just taking the product and filtering and giving to us. It is wine and beer. Whereas whiskey, brandy, and rum are produced by distillation of the fermented product. I'm not included any single term from outside. This is what is there in your book only. I have straightaway used. So these are something about that. And second one is penicillin. For this, I'll show this diagram. So here you can see the soldiers are treated with penicillin in Cold War. So something is there to discuss with penicillin. So if you look about antibiotic, anti means against, and what do you mean by bios?
Mm,
Life. Life. Bios means life. Against life means not for against us. So what is antibiotic? Antibiotics are nothing but these are some chemical substances produced by a one microbe to kill or stop the growth of another microbe. That means here I'm not going to discuss with human being. It is interaction between one microbe and another microbe, where one microbe is able to produce some chemical, and this chemical is able to kill another microbe. One microbe will produce some chemical that will kill the another microbe. For example, I have a fungus called a Penicillium notatum, children. Here it is a fungi. Here it is actually a culture plate where we have the bacteria. You can see this is the normal pathway which I'm going to draw and show you. This is a normal pathway of streak plate which we have made. But now, when in the culture medium, if I have drawn a farm, if I have just accidentally, if I drop a penicillin containing medium, just small drop, you can see this penicillin is remaining as such. The penicillin is not grown much, but surrounding this area, if this penicillin is here, it's okay. But you can see surrounding this penicillin, some secretion is produced by the penicillin. This is the penicillin fungi penicillin. This fungi penicillium is able to produce one chemical which is shown here. So when the chemicals are produced by them, this chemical is stopping the growth of a microbe. So the chemical produced by Penicillium notatum is able to kill or stop the growth of a bacterium. So this is a small history of penicillin. So we look about penicillin. Lot of demonstration and all is there. So we look about an antibiotic. Antibiotic here is Penicillium notatum is able to give some chemical which stop the growth of staphylococci. What the chemical called as? That chemical is only is called as penicillin. Penicillium notatum, a fungi, will produce a chemical called penicillin. This penicillin will kill the growth of microbes. So the here chemical here is penicillin. So antibiotics are nothing but a chemical substances which are produced by some microbes which will kill or retard the growth of other disease causing microbes. And the first antibiotic only by a chance. I told you here purposely I didn't add. It was by a chance. Accidentally what happened? We had some person called Alexander Fleming and Ernest Chain and Florey. So they were working with Staphylococcus bacterium, and they threw the Staphylococcus bacterium colonies to wash in the culture. So after to wash, they just throw it in the sink. In that, the student of Alexander Fleming accidentally he dropped the penicillium growing colony into the medium. Next day, when he came and saw, he was able to see that the penicillin notatum was thrown into the medium where Staphylococcus was growing. The Staphylococcus growth was stopped. Then only they made lot of research on that. It was an accidental one by a chance. So Alexander Fleming, while working with Staphylococcus bacterium, once observed that a mold growing in one of the unwashed culture plates around which Staphylococcus could not grow. So Staphylococcus was not able to grow. He found out that it was due to a chemical produced by the mold. He named it as penicillin. From a mold called Penicillium notatum, they discovered a chemical called penicillin, and that is used in antibiotic. But the fullest efficiency was brought by Ernest Chain and Howard Florey, and they all shared the Nobel Prize. So, what are the properties of an antibiotic? That is very important. So, the properties of an antibiotic are number one, an antibiotic should be a broad spectrum, but here it is a narrow spectrum because it was working against staphylococci. Generally, it should be a broad spectrum. It should have the ability to act on various pathogenic microbes. Second, it should check the development of resistant varieties of pathogenic microorganisms because soon they may gain resistance by natural selection theory. About that in evolution, I'll be teaching you. An antibiotic molecule should attack and disrupt a specific vital link in the metabolism of pathogenic, and they must stop the metabolic growth of the disease causing microbes. It should not cause any harm to human beings. It should only kill the growth of another microbes. Action of antibiotic molecule should be specific on pathogen in critical cases. It should not disturb the other beneficial microorganisms. So, for example, I'll tell you an antibiotic called as common antibiotic which you are using. For example, polymyxin is actually polymyxin B is an antibiotic. It's not in your book. It's obtained from Bacillus polymyxia. And if you look about erythromycin, okay, commonly we use for a throat infection, erythromycin, that is obtained from a bacterium called Streptomyces erythraeus. So everything, even amoxicillin also an antibiotic, we are extracting only from the bacterium. I mean, sorry, microbes. The third topic in industrial purpose is microbes as for extraction of acids, where we have a fungus called Aspergillus niger, a fungus which helps in production of citric acid in industrial purpose. Acetobacter aceti, a bacterium for acetic acid production. Clostridium botulicum, a bacterium for production of butyric acid. Lactobacillus delbrueckii for lactic acid production. Saccharomyces cerevisiae for ethanol production. Not only this, children, see, we all are having food in our home, right? And we always use sambar and all. The first thing to make sambar is we add oil in everything. Then we add those protein containing and molecules and all we'll be adding. The first thing is, and we'll be using only oil, means lipids. Therefore, when we want to wash, that lipid has to be removed off because very because it will make stains on your clothes. So for that, in laundry, in detergent, for detergent we use a lot of formula called protease, RNAs, and all these chemicals will be added. Next up, we're going to add an enzyme called lipase. Lipase will is mostly used in detergents, and because of that only the fat molecules or the oil droplets can be easily removed when your mothers wash the clothes. And we look about an important thing, children, here. Your mom will make pickle, and you can go to market and buy the pickle, which will be tasty actually. Mom's only will be mostly tasty, but which bottle will be very attractive? One which is made in home or one which is made in industries?
Industries.
Industries, it will become so shiny, the bottle looks so shiny clear. Amma pananga, what will be there? The bottle will be a little bit dull, but taste cannot be compromised. At the same time, when you go to industry first, taste is unique for them, but thing is the bottle will be uptake will be very clear. So there we use an enzyme called a protease and pectinase. There's pectinase and protease enzymes which is used in bottle juices, so bottle juice obtains so very clear. And this is an injection called streptokinase, a type of product obtained from streptococcus, which is a bacterium. This is used to remove the blood clots. So people who have blood clots in their blood vessels, for them they use a streptokinase enzyme. So streptokinase is obtained from a bacterium called as streptococcus, used to remove the blood clots. Only three enzymes are there. I'll just with that I'll finish it off. Cyclosporin A. This is a cyclosporin culture, and it is a fungus called Trichoderma polysporum. So cyclosporin A is obtained from a fungus called Trichoderma polysporum. It is used for immunosuppressive agent. Why? Because a person who have gotten organ transplantation, for him the new organ or the foreign particle, or we can say any foreign particle enters in your body, it will be rejected by your immune system by cell mediated immunity. When your immune system is suppressed initially, the organ can be accepted. So for a patient for whom, if you want to make a successful organ transplantation with all the 26 parameters of blood matching, tissue matching, everything is proper, still there is a chances that the foreign organ can be rejected by your body. For that, we use a drug called cyclosporin A, obtained from a fungi called Trichoderma polysporum. It is used as an immunosuppressive agent. The next is called statin. So statin is used as a blood cholesterol lowering agent obtained from a yeast called Monascus purpureus. So with this, I'm going to stop there, and sewage treatment we're going to see in the next class. Okay, children, thank you very much.